right, we're gonna go ahead and get started. Yesterday we were working with fruit flies, right? I think all of you were in that group yesterday. What were we doing with the fruit flies yesterday? Yeah, go ahead, Garth.
We put them on ice to see their reaction and stuff.
Why did we use fruit flies and not a person or a mouse?
Because they're cheap.
They're cheap, yeah. We can get a lot of them. They're easy to handle for the most part, unless they flew out of your vial, right? We're going to look at another variation where we're looking at fruit flies as a model. And for this one, we're going to be looking at nervous system defects. So, do you have a nervous system? Yes. What's the function of your nervous system? Nervous system. Yeah, what do you think?
Doesn't it make you nervous?
It's part of how you react to things. So if you are afraid of spiders or you're anxious about a test, that would make you nervous. It's your body reacting to that, right? But your nervous system's got how you sense and process information. So your sense of touch, hearing, taste, smell, all of those things. Yeah, your body's going to sense that and process that information.
Process that.
So
You can feel somebody touch your arm or any part of your body.
And just like every system that you have, stuff can go wrong with it. You can have diseases, mutations that pop up in your life. Some you develop in your lifetime, some you inherit, they're genetic. We're going to be looking at some of these today. So on your sheet, on the top, you have that box where it has a silhouette of a person and then a mouse. You guys see that on the top page? On that page, on that table, we're gonna look at the person first. You're gonna make, absolutely. You're gonna make observations about what you see. Work. There we go. You're going to make observations about what you see in the video. And then you're going to make, after we do observations, you're going to make inferences or questions that relate to your observations. What's the difference? What is an observation again? Yes, Ro?
Like a voltage or something.
Say that again?
Like a voltage of something.
Okay, so what you see, okay, what you hear going on. Are you trying to explain what that is in an observation? Or are you just saying it how it is, saying what you see? Saying what you see, right? So that's going in your observation box. And then inferences and questions is going to be you kind of doing some interpretation or questions that you may develop based on those observations. We'll stick with the observations first. So quick video. I want you to pay attention to this little boy's hands. And then what is he putting together here? A puzzle, right? I don't think sound is critical. So you want to jot down some observations that you're making in that box. So what are some observations you guys have? What do you see? What do you see going on? Girls on this side, anything? Do you notice anything with him putting together his puzzle?
Do you see anything different from what you usually would see?
Yeah. His hands are shaky. Yeah, that's one.
Good.
He's being really careful and picking and choosing.
Okay, so he's being very careful.
Yes, sir.
He has brown hair.
He does have brown hair. That is a very good observation, but focus on anything about how he's putting together the pencil. Not the pencil, the puzzle. What am I saying? Any observations? Yeah.
His hands are tensing up.
Hands are tensing. Look at his right and his left hand. Is he able to ever move them independently of each other? When you move your hands, do you have to move them both at the same time, or are you able to control them? Watch his hands. Is he able to control them independent of each other, or are his movements mirrored or duplicated on the other hand? You notice how he's grasping the puzzle piece with his left hand? What is he doing with his right hand?
Grabbing the other.
He's acting like he's grabbing onto the puzzle piece, right? Is that how it works with you? When you grab a pencil, does your other finger grab a pencil at the same time? What happens gets grabbing it? No. Okay, so something's going on. So in your observations, all your observations were good. Okay. I also want you to add a piece that says something how his movements are, he's not able to control both hands.
So when he grabs the puzzle pieces that are in front of his hand.
This book is famous.
Now, what would be a question you would have based on those observations we just made? Yeah.
Why is he doing that?
Okay, why can't he control his hands independently, or why is he doing that? It's a good question. All right. What might not be working in his body that would be responsible for those movements? Garth?
His nervous system.
His nervous system, something may be up, right? With his nervous system, something may not be firing right. Good. Let's move on. All right, so now a mouse.
A mouse.
Watch how the mice are moving. So that's a normal mouse. Pretty standard. Okay, here are the ones that have the problems. So you're going to make observations about the mice, what you see. Try to focus on how it moves. What observations do you have about how the mouse is moving? Yeah, in the pink.
When he walks with his front paw on the other side, both of them go at the same time.
Both of them go at the same time. How did that compare to the normal mouse we saw at the beginning? How was the normal mouse moving at the start?
Justin, go ahead.
I was like walking normally.
Walking normally, right? One foot in front of the other. And then the other examples, kind of looked like he was hopping, right?
Look like a bunny.
Looked like a bunny. Okay, that was because the legs are moving at the same time.
Nervous system.
Yeah, so make sure you write some observations regarding how it's moving and that it was not able to control both feet at the same time, right? Isn't that something similar to what we just saw with the boy, little boy?
Yeah.
Yeah. And then I think one of you guys already mentioned an inference that something must be going on with the nervous system. Okay, you think something's going on with the nervous system again? So that could be a good point to mention there. So, would it be easier, more ethical to study nervous system abnormalities in humans or in mice? And why do you think that? Yes, sir.
Mice because they're not as there's a lot of them and they're cheap.
There's a lot of them and they're cheap.
But they're not as cheap as flies.
Not as cheap as flies. Anything to add?
I would say humans
Okay.
because I feel like it's more important.
More important, okay. Alright. Did we see a similar kind of behavior in both the mouse and the human, right?
Yeah.
Similar kind of stuff. I agree with you in that if we looked at a human, we'd probably get kind of immediate information that we could apply to how we work, right? But if I wanted to do a lot of experiments and have a room full of subjects that I can do experiments on, is it going to be easier to get a room full of people or a room full of mice? Mice, right? We can get a lot of cages, a lot of mice, do a lot of experiments.
And humans are just
Plus, the other thing you want to keep in mind too is a lot of times when they're doing studies, you're doing studies to see if something is going to help. But you never know if it's actually going to help the organism or it could potentially make them worse.
Yes.
Or it could kill them. So if you're using humans and you take something that's very untested and try it on a human, it could kill the person. So our goal with medicine is always to treat the person to make them feel better, but not to kill them. That's why the way it is in our country, there's a lot of tests that have to go on before they can even get to do trials, human trials. And then when they do human trials with medicine, it has to be voluntary. Or sometimes they'll offer to pay people for it, but it's not something that they can just come to your parents and say, I need a 12-year-old boy? 11-year-old boy. So we decided we want your son because we want to see how this medicine works on him. It doesn't work like that. And there's a lot of different things that people have to take care of, basically just to protect everybody's health and to make it better for everybody.
Someone told me, I forget who, but I feel like someone told me Hitler did like human experimentation. And
He did.
He advanced for battlefield 50 years.
That I don't know about, but I do know that, yes, Hitler had scientists doing human experimentation.
Yeah. Frowned upon. Takes a lot of trials before you get to that point. Now, we are all experts on this guy, Drosophila melgaster, the fruit fly, also used as a model organism, right? So while we can get a room full of mice and have maybe 50 mice in cages, we can get hundreds and thousands of fruit flies in the same space, right? Another model organism.
Can you flip back a minute? Kayla, based on what you showed us, the Drosophila thing, that would be a male, correct?
Yeah.
I was going to say,
Oh, interesting.
before lunch, we were looking at how you can tell the difference between females and males, and because of the dark abdomen at the end, that's how you can tell it's a male.
Huh.
One of the ways.
The more you know about that. So why do we use Drosophila or fruit flies as a model organism for looking at humans? Well, first, they're an animal, right? Last time I checked, you were all animals, yes?
Yeah.
Yes, you agree? If we look at genes, so when you hear the word genes, that's just going to be parts of your DNA that do something important for you, so they make code for proteins. So the DNA that you have in common. So we are in the center, right? Our closest relative is going to be a chimpanzee. What percent of our genes do we have in common with a chimp? 98%, that's a lot, right? Do you see fruit fly up there? What percent do we have in common with fruit flies? Yes?
44%.
44, right? So it's a good amount, right? It kind of, and then above that is going to be the mouse with 92, right? So the closer you get in terms of relatedness to people, the more similarities that you see with the DNA, right? And then a plant is all the way over here with 18% similarities, right? 18% of your DNA is kept in a plant, right? Anything to add on that one? Anything to add on this one? You good?
No, I'm good.
Okay. So if we're looking at similarities with humans and fruit flies, right? Fruit flies are animals with, they show a head region just like people do. They have, feel free to add anything, fruit fly expert, okay? Fruit flies have a head just like we do, and the head is just kind of like a clustering of sensory organs. They have their eyes there. They have antenna. Yes, they have antenna. We don't have antenna, but we have ears. They have their brain, mouth parts, all in their head, just like you do, right? Some differences. And they also show a brain, as well as our brain, obviously. That's going to be the fruit fly brain here. We say that humans and fruit flies show bilateral symmetry. What does it mean to show bilateral symmetry? Yes?
The way our body parts sort of fit together, like the positioning is...
Positioning, okay. You want to add on to that? You good?
The brain.
Okay, so the brain shows bilateral symmetry, right?
Organs are symmetrical.
Yeah. So if I would take you and then fold you in half, right down the middle, would your halves match up pretty evenly, or would you be like all off and?
Halves match up.
Yeah, the halves match up pretty easily, right? So it's an important trait that a lot that we share by that bilateral symmetry with the fruit fly. Not all animals show bilateral symmetry, so it's a really important characteristic that we share with fruit flies, and we see our brains match in that they both show kind of like that. You can put a line down the center, and you can fold the brain in half, and then it looks like it's a mirror image of each other, right?
Almost.
I mean, it's not 100%, but it's almost there, right? Okay. We can look at our nerve cords here. So we have the fly nerve cord on the left, human spinal cord on the right. Any similarities you notice between the two? You're a fruit fly expert today, aren't you? Let me go with her quick and then I'll come back to you. Yes?
So they have different...
Okay, yeah, so it's kind of sectioned, right? They have multiple components. What are these little spaghetti-like projections coming out of the central cord here? What do you think those are? Yeah, I heard it. What is it?
Nerve.
Nerves, right? Nerves are just the specialized cells involved in a nervous system, right? So they have nerves coming off the central fly nerve cord, right? We don't see that with the human spinal cord because it's not, because this is taken from, those parts are not visible here, but we can imagine our nerves coming off of these kind of intersection points here. So that is going to be a similarity between the two. Are they the same size?
They're about as big as the air as the finer cord is about as big as the fly, and the human spinal cord is about as human.
Right. So they're about 1 millimeter, 1 meter. Okay. So size difference. They're different size organisms, so they're going to be different. What about the stuff that covers the human spinal cord? What does the human spinal cord have surrounding it that the fly does not?
Everybody take your left hand and simply reach around the back of your neck and feel at the base of your neck. What can you feel there?
Bone.
Bone.
Yeah, so-
Exactly.
We're going to have bones surrounding ours, while fruit flies do not have internal skeleton. They are arthropods. They have the external kind of external shell. So they don't have bones like that. These are just around the same position. So the life cycle, yesterday we were working with the adult. Here, adult female, adult male. Those are the ones with wings, obviously, but the entire life cycle does not always look like the fly that we see, and that fruit flies actually spend a good chunk of their life as little larvae or little bitty worm-like critters that if we look in those vials that we looked at yesterday, you'll see kind of them kind of crawling around in the bottom. They're really tiny. And then here we have a pupa, right? And the pupa is just the point where they transform from the pupa into the adult. Why do you think we want to study the nervous system in embryos instead of adults? So we're going to be looking at embryos. Garth, what do you think?
To see if it grows like the way it should.
That's a good point, yeah. To see if it has any like developmental impacts. So yeah, that's an important point, yeah.
See how it grows.
See how it grows, right? Okay. Anything else? Any other point you think?
See change.
About change, okay. I don't know what else we would do with embryos.
Well, if you think about, there's a whole area study for human anatomy in neural tube defects and how the spinal cord develops in humans, that if we watch the embryos of fruit flies, for example, we can actually see easier like where it goes wrong and then try to determine what we could do to get it back on track if there's anything, or how we could fix it. And for example, there's something called anencephaly in humans, where as a baby develops, it will have only the very lowest part of the brain or the brainstem develop, and basically from the ears up, there's nothing. And that's considered a neural tube defect because it's when the nervous system's developing and just a whole part of it's missing. And those babies can't survive. So that would be something that we'd want to figure out how to identify or possibly how to fix in the future if there is a fix. Just because we can figure out where things are to identify things doesn't always mean we're capable of fixing them. And when we're talking about brain development especially, if you're missing such a huge chunk of it, it may not be something we can ever fix or figure out how to fix.
So when we're looking at these neural defects, we can create fruit fly lines or fruit fly kind of colonies where we can cause them to develop that mutation or that condition that we're interested in studying. So think back to that nervous system defect. We can cause that condition to occur in fruit flies, right? We can cause it to occur in fruit flies, and then we can use those fruit flies in our studies. So we have our so when I see that word wild type, that's going to mean average Joe fruit fly, normal fruit fly. Okay? Wild type means it's the one you expect to find in the wild. And then this one we treat with a mutagen, and a mutagen is just a chemical that causes DNA changes, right? So we cross a normal fruit fly with one that was treated with mutations, and then we produce a normal fruit fly again, and then we have a variety of males with different mutations. And what scientists can do is they can screen all these mutant fruit flies to find the one that has the nervous system defect that they want to study. So if we're interested in looking at these nervous system defects, we can screen these fruit flies, find the one that has the nervous system defect, and then mate that one, which is here, with our normal female. And that would then produce a bunch of individuals that carry or hold on to that have that nervous system defect gene in their DNA. Okay, so what happened is we got these cultures from scientists doing this research. They created these lines for us. They caused this mutation, and then they selected for the males that have the mutation, and then we are left with their offspring here. They cross these individuals, and now we are left with their offspring as little larvae. Now, on your sheet, on the bottom of the first page, I went through these terms. I mentioned what wild type was. So what is wild type? What does a wild type fly mean? Normal fly. So next to wild type, you want to write normal on the bottom of the first page.
And just so you know, that's why I was looking on my computer real quick. The vials we use that have a 0 are our controlled ones. The ones with an X are our mutant ones when we go over to the lab.
So wild type is going to be our normal flies. And then who remembers, what do we add to the mutagen treated male? What do we do to that male?
Change the DNA
Yeah, how do I change it? Yes, sir.
by adding a mate.
Adding a what?
A mate.
Adding a mate, so the mate's gonna produce offspring. I change the DNA with a chemical of some sort, right? So a mutagen treated male is a male that's had its DNA changed. And then on C, that plus and then an A. Can you turn the light on? I can do
Yeah.
that on the square now. The plus and the A, I want you to write next to that one carrier. I'm going to tell you what a carrier is next. So next to, for C, it says plus and A, you're going to write carrier. C-A-R-R-I-E-R.
I think I know what it is.
What is a carrier?
A carrier is a example of fly with the mutation that you gave it, and then it carries it to other flies that have.
What do you mean it carries it to other flies?
It has the mutation on it, and then when it mates with another fly, the fly will.
Does it show the mutation?
No,
Why not?
because it's DNA.
I like that. That's good. So if we look at plus A, that plus gene means it's normal. And then the A is mutant. So it has one copy of a gene that's normal, one copy of a gene that's mutant. Overall, this one's going to be a carrier, and it's going to act normal, right? Kind of what you said, yeah? It's a carrier, it's acting normal, has a normal copy of the gene, and a mutant one. That normal one, we can say, is dominant over the mutant one. Now, if this fly mates, what could happen to that mutant copy of the gene?
It could become mutant.
What would happen to that mutant copy of the gene if I were to mate that fly with another fly?
It will transfer to the other fly.
Not the other fly, but who is he going to pass on that mutant gene to? Who do parents pass their genes on to?
Their offspring.
Their offspring, right? So he's going to have a chance of passing that mutant gene onto the offspring. We got one more thing to do, and then we're going to go in the lab. So flip to the second page on the top. So what the person in the lab who sent us these flies did is they crossed two carrier flies. So they took one fly who was plus A, and they crossed that fly with another plus A. So they both look normal because they have the normal gene. So they cross both of them. And then they're going to produce baby flies, kids, right? And I think you guys are going into 6. So we can use something called a Punnett square. Has anyone done a Punnett square before? No? You've done Punnett squares?
Probably not.
Probably not. So we're going to do something called a Punnett square. You're going to do this in 7th grade, but what Punnett squares allow us to do is we can take a mating between these two flies and we can predict what their kids are going to look like. Kind of give us an idea of what we would expect. So we can set up this Punnett square, and you have one already set up on yours, on your handout. Right here on this, it's like that grid system on your second page. So when I do the Punnett square, and you guys can follow along with yours, I just take one parent up here, one parent on the side, and then we can use this then to fill in and predict what types of offspring we will get in this sort of cross. So when we do our cross, we can fill in the top left box, we get plus plus, bottom left, we get What's bothering you?
No, you're good.
The bottom left, we get plus A. Top right is plus A. Bottom right is AA. Now, when we do this cross, what are these flies going to look like? Are they going to be normal or mutants?
Normal.
Why are they going to be normal?
Because the pluses mean normal.
Pluses mean normal, and that's the only thing they have. Ladies over here, this one, plus A. Are they normal or mutants?
Mutants,
So they have the mutant gene, right?
and they also have the normal gene.
But how do they look?
Which one dominates, the normal or the
The
mutant?
normal.
Normal. So these ones are gonna look normal, right? And then same down here, normal. What are these guys gonna look like?
Mutants.
Mutants, right?
Now, let me just show you what I was going to show you guys, okay? What we do in 8th grade, Mr. Esser and I, when we do stuff together, oftentimes he'll do an explanation, and if I have it a different way that I do it, then I'll hop in. So like when I tell students about Punnett squares, my thing has always been, how many of you know how to do a tic-tac-toe board? Some of you guys have known since you're this high, right? So you can draw a tic-tac-toe board, okay? Your parents, you would put on the top and the side, just like Mr. Moraski did over here, because they're floating down here. Then you bring your fingers together. What do I have up here? What do I have over here? I bring them together. And you'll see we get exactly the same results, only I just did it a little bit differently. Okay, so it's the same exact thing here, and then if you look on your paper has
Closed
this
box.
as a closed box. Okay, is any one of those more right than the other? No, you can do all three ways. Okay, but this is something one of my professors many years ago showed us, and she always said tic-tac-toe is something you guys have always known pretty much since you can remember things. Because that's one of the first games most students learn. So it's another easy way to do that. And you always want to keep in mind that you get one allele from each parent, because these things are called alleles. So you get one allele from each parent. So you're always going to have two alleles for every trait.
Now, looking to the right of your box, it says, what percent of the offspring will be mutant? So we said one out of how many total possibilities are there? 4. So for A, it's going to be 1 out of 4, or what is 1 out of 4 as a percent? Say it out loud, someone.
25%.
25%. The 25% will be A. 25% of the offspring embryos will be mutant. And then what percent of the offspring will look normal or wild type? 75. So it's these 3 are going to look normal. So B is going to be 3 out of 4 or 75%. So what happens? Scientists did this cross for us. Then we produce these mutants and normal type flies. Let's skip the next one. I'm going to show you what you guys are going to see in the lab, and then we'll go in there. So what's going to happen, we have vials, but we're not going to see flies flying around, right? Why wouldn't, that doesn't really bother us because we want to work with the larvae, yeah? So they're all going to be vials of larvae. They're not looking like they're going to move a lot, but that's okay. So what we're going to be doing is we're going to get our vials and we're going to pour in some sucrose solution. What is sucrose? They do eat it, right? But sucrose is going to be table sugar. Okay, it's a chemical name for table sugar, stuff you put in your coffee. Well, you don't drink coffee, my coffee, but it's stuff that you like. Sweet sucrose is just a chemical name for it. So we're going to put sucrose solution into the vials with the larva. Okay, and what's going to happen? See, it kind of looks like murky water. We're going to have the larvae are going to float. Apparently, fruit fly larvae are buoyant. Who would have known, right? They're buoyant, they float. Why do we use sugar water and not pure water or salt water? Why do we use sugar water?
So they don't it could stick to the ground.
They
And
could.
then the sugar could come in and push them out.
Let's do the salt water. When you go to the ocean, what happens to your skin?
It prunes up.
Prunes up. Why does it prune up a lot?
Because of the water getting into your skin.
Is it water getting into your skin?
Oh, no, water going over your skin.
Over your skin? Think of a raisin, right? A raisin is a grape. Does the water go into the grape or leave the grape to turn into a raisin?
Leave it.
Leave the grape, right? Doesn't like a grape raisin skin kind of resemble your fingertips after you go in the water, right? So did water enter your skin or leave your skin when you go to the ocean? Leave, right? So what happens is that the salt in the water draws out the water in your skin, resulting in the pruny kind of like wrinkles in your skin. Now, if we put very tiny fruit fly babies in salt water, are those fruit fly babies going to be happy? No, they're going to dry up and die, right? The water is going to be drawn out of them. Similarly, if we use distilled water, okay, I'm going to tell you with distilled water, that's pure water, that water is going to move into the fruit flies and the fruit fly is going to explode like a balloon, right? Water's going to go into it, it's going to explode. That's a whole complicated biology and chemistry with that. Just trust me with that pure water is going to cause them to explode. Not good. So we're going to use sugar water. They're going to float, and what you're going to do is you're going to use a paintbrush to pick up 3 to 4 larvae. So we're going to paint them. There's the paintbrush going in. Larvae look like little worms that move. And then you're going to place the larva on a piece of photo paper, and then you're going to paint the larva with food coloring, right? And then remember, the point of this activity is we're looking at that nervous system mutation. And think of the rat. It affected how it moved, right? If these larvae had a nervous system mutation, where do we would expect them to have issues with their movement, right? So what will happen to the larvae who have the mutation? What is the dye going to allow us to do? What is that going to allow us to see? Yes?
It's sort of going to create a trail.
It's going to create a trail, right? And we can compare what the normal fruit fly larva looks like and compare it to the one that's mutated, right? And this also looks like a really cool spin art with fruit fly larva, okay? That's also something that's really cool with this. So we're going to go into the lab. So take your things, pencil, paper. We didn't get to all the packet, that's okay.